So I have a question for you. Why do two people who eat similar diets often have very different gut microbiomes? Well, a new major study in the Journal of Nature suggests that part of the answer lies in our genes. So yesterday I talked about how a new taxa of microbes seems to play a beneficial role in preventing disease in your gut microbiota. Well, today I'm going to talk a little bit about how genes may play a role in the diversity of the microbiota that people have. So this recent paper is from the Norwegian Hunt study and is one of the largest and most rigorous investigations of how host genetics and the gut microbiota interplay between each other. In this study, the researchers used shotgun method genomic sequencing from over 12,000 individuals and replicated their findings in multiple independent cohorts, allowing them to identify genetic associations that have been difficult to reproduce in previous microbiome studies. What's really interesting about this study is that it identified 12 robust host gene microbiome associations across 6 genomic loci, meaning what they found essentially was 6 genomic regions in the body that seem to dictate what particular microbes and how they function are in the microbiota of your gut. Some of the strongest signals they found involve the gene LCT, which is responsible for lactase persistence, the genes ABO and FUT2, which influence the glycan structures present on mucosal surfaces, infect the gut environment, and what is excreted in the intestines. So one of the cleanest examples they show is the LCT locus. Individuals carrying variants associated with lactose non-persistence leave more lactose unabsorbed in the gut. This creates a selective advantage for lactose-fermenting bacteria, particularly members of the genus Bifidobacterium, and this finding therefore provides a mechanistic example of how host genetics can alter nutrient availability and in turn shape the ecology of the microbiome. Also super interesting is that through the use of Mendelian randomization analysis, the authors were also able to show evidence that higher body mass index may causally influence aspects of microbiome composition. This suggests that the relationship between obesity and the microbiome is bidirectional, with metabolic state affecting the microbiota as well as vice versa. The broad overarching significance of this work is that it strengthens the ecological model of host and microbiome interactions, meaning that human genes help determine nutrient availability, mucus glycan specificity, and immune pressures within the gut, creating specific niches that favor particular microbial communities and their functions. In essence, or in other words, our microbiome is shaped not only by what we consume and the environment we live in, but also by the inherited genes that we get and we pass on from generation to generation.